to have any notes or anything like that. How did you guys feel about your exam? I know it was a rough week and it was a hard unit. That's why I put up the practice exam and opened it for a few extra hours. But hopefully you guys were okay to get through it. I know Dominique's sweating it. Yes, I totally agree. I was sweating it too. So I will grade those this week and get them back to you. I know that was a tough unit, but you guys persevered. You always do. You will be amazing. So let's go ahead and move on to the next one. That's behind us, and we're starting out fresh. Okay, so today we're going to talk about pathogenicity, and what we're really talking about in pathogenicity is how we react to a pathogen, which was really mostly our immune system, and then what is it that the pathogen is doing that we are actually reacting to. So what kind of toxins are they producing? How are these toxins interacting with our bodies? What's happening? So that's what pathogenicity is all about. And of course, just like every other unit, we usually have to start with our normal microbiota, and we're also going to go through some vocabulary. So our normal microbes do play a role in protecting us, and we talked about it a little bit in the immune chapter unit. However, we didn't go over the different relationships. So our normal microbiota normally play very protective roles for us, but our normal flora also can become pathogenic. So we call these opportunistic. The roles that they play, of course, are antagonistic. So our normal microbiota are oftentimes very antagonistic towards transient or pathogens that may be entering into the system on a temporary basis. We have mutualistic relationships with some of our flora, where kind of you scratch my back, I'll scratch yours type of thing, where we provide those organisms with this great place to live with lots of food and nutrients and water and such, and in turn they provide us with something that we cannot necessarily make ourselves, some kind of metabolite, like vitamin K, for example, is one that we can absorb a lot of vitamin K, particularly through like dark leafy greens, but we don't process it well. So microorganisms actually process precursors to the vitamin K, and then the metabolic waste is vitamin K that we can absorb. Parasitism is another relationship. However, it's not protective, it's pathogenic. And this is when our own microbiota can turn against us. Perhaps we introduce Staph aureus on our skin to a new environment by cutting our skin, and now they're introduced to a new nutrient source. This changes their metabolism and initiates production of certain toxins. So we can sometimes have many of our own infections, and very common infections are due to our own normal microbiota moving from a mutualistic or commensal relationship to a parasitic or even antagonistic towards us. And then commensalism is kind of what we have with some of the flora maybe on our skin, where it doesn't really serve us a big purpose. We provide it with a place to live. It's kind of like it's there, but it doesn't really do us any harm. So we just allow it to be there. Now, keep in mind that the population and diversity of your normal microbiota is going to change constantly. It is forever changing. If you decide to go on a diet or go keto, that's going to completely change the flora in your gastrointestinal tract. It's going to change the diversity and makeup of it. If you get older, if you move to a new area, a new part of the country or a new part of the world, your diet is going to start causing changes, and all kinds of different things can cause variations in your microbiota makeup. I gotta find my little tab here. There we go. So that brings us to the idea of pathogenicity. And a pathogen is anything that's going to cause a disease. So we just spent, what, 4 weeks looking at how we respond, excuse me, to different pathogens. But anything that actually evokes our immune response would be a pathogen. Allergens are a little bit different. They are still antigens because they evoke an immune response, but they don't cause disease, so to speak. They're just considered a little bit different. An opportunistic pathogen is a member of our own normal microbiota that's going to cause some kind of disease in the host when it's our defenses are compromised, or there's been some sort of change in the makeup of our microbiota, those sorts of things. Virulence and infection. So virulence is sometimes used interchangeably with pathogenic. How pathogenic is something? What they're really asking in that question is how virulent is it? How intense or to what degree is this disease going to get? We're looking at COVID-19 right now, and COVID-19 is a coronavirus, say very similar to SARS. Now, SARS did not cause a worldwide pandemic, and SARS, as far as we know right now, had about a 10% mortality rate, which is really high. SARS was a very deadly respiratory disease. But SARS was harder to get. Its transmissibility was much more difficult than we see with COVID-19. And so COVID-19 is super, super contagious. And so the virulence of SARS was greater in terms of a mortality rate. However, the transmissibility in COVID-19 is much greater. And of course, because it's a novel coronavirus, there's no immunity to it. And that's why so many people are getting sick. And infection is just simply a term used to a general term for whether it's a virus, a bacteria, toxic, or if it's a systemic or localized infection. So in diseases, we've looked at these terms before, but I just want to reiterate them because pathogenicity is part of these principles of disease. And so if a disease is communicable, it's contagious. So COVID-19 is a communicable disease. And so this will spread from host to host. And whether it's through droplet transmission, aerosolized transmission, touch, fomite transition, those sorts of things, it's still considered communicable because it travels from host to host. There's no vector, there's no intermediary, and there's no environmental reservoir aside from the hosts themselves. And in a course of disease, we have incubation, illness, and convalescence. And this is your traditional, right? So these three factors here are your traditional acute disease. And that's what we think of when we have a cold or the flu or some sort of disease. We get exposed and there's an incubation period. We then get sick. That's our illness phase, our immune system kicking in and that sort of thing. And then in recovery, when we're in recovery, we're in a convalescence phase. But when we start looking at other types of diseases, such as those that are chronic or latent, remember, chronic diseases, they just persist over a really long time. You never really, there's no convalescence. You just don't ever really get better. You'll usually go through an acute phase and then come out of that acute phase and then just kind of stay at a low level. You might get better a little bit and then it just eventually will come back up. So these chronic phases, these illnesses last over a really long time. That doesn't mean that it necessarily leads to death. It just means that this is an illness that will take a really long time to get over. And then we have latent diseases, and latent diseases are those that we have those recurrence. So the person goes through an acute phase, and then they'll go through several phases again, and oftentimes they'll go through a final acute phase that results in death. So you have acute illnesses, chronic illnesses, and latent. What causes a disease? How do we determine the cause of a disease? And this is determined using what's known as Cochise Postulates. So Koch was a kind of, he was a science, he just did research, he was a microbiologist, and he figured out, he actually did more veterinary medicine, and he was looking at a bovine version of tuberculosis, or a cow version of TB, a wasting disease in cattle. And he discovered that there was a way to systematically determine what organism caused what disease. And so he came up with what is referred to as a set of rules known as Cochise Postulates. And these postulates are still used today to determine and to, I guess, solidify the fact that a specific organism or virus or pathogen causes a specific disease. So the first thing you have to do when you're trying to figure this out is you have to see the organism, or the pathogen, we should say, present in every case of the disease. So every time someone is suspected of COVID, they're being tested for it. So we have to make sure that that's what they're actually suffering from. We have to be able to grow the organism in pure culture from a diseased host. Now this would only work in bacteria. If we're looking at viruses, we would have to use a cell culture. So we would have to use a cell culture for this. And then if we reinfect a healthy host with the same pathogen, then we should see that disease occur in the new healthy host. They should have the same disease and the same signs and symptoms. And then, of course, we look for the organism from the newly infected host. We need to be able to isolate it from the newly infected host, and it should match the organism that was grown from the original ill host. So this was all done back in the 1800s, and it was ethically okay to infect animals and hosts and those sorts of things. And so that's what Koch built his postulates on. The problem we have is that it doesn't work well for viruses, and it doesn't work well for bacteria that are intracellular, organisms that require a cell in order to grow. So for viruses and intracellular obligates, they have to use cell cultures, and they have to take Koch's postulates and modify them. So in the modified postulates, they're going to use either the production of a toxin or they're going to use genetic material. Now, we're looking at this COVID-19. In COVID-19, they have sequenced the genome and they use PCR. We looked at PCR when we did biotechnology. So the confirmatory test right now for COVID-19 is a PCR test. And one of the problems they had when that PCR test was rolled out were the primers that they were using. And the primers were not working well, and they were getting a lot of false positives. So that's why the test had to be recalled and redone. The virulence factor gene, or the product of that gene, we can look to see if the product of a specific gene is present. Instead of looking for the genome itself, we can look for whatever it is that the gene produces. We can go into that organism, let's say it's a virus. We then isolate that genetic material, we mutate it purposely, and then we're looking at mutation of a gene. So this is known as point mutation. So a gene is selected in the virus or in the bacteria that we're looking at. We select a specific gene. We insert, using biotechnology, we insert something into the middle of that gene. This is kind of like we did in lab when we inserted a gene into E. coli. So here they would insert the gene, insert a small piece of DNA in the middle of this gene to give it a loss of function. So now this gene no longer functions. If it doesn't function, we then look to see if the product of that gene is still present. If it's not, then we know that we mutated the correct gene. We then remove the mutation from that line of organism, and if we reverse the mutation and the function is restored, then we know what that gene does, and we know that gene is associated with the pathogen. So we can then look for that gene specifically using PCR. So we know that that would be a virus-specific gene. I know this sounds kind of complicated, but we're looking to make sure that whatever it is we're going to use to test with is virus specific. We don't want to test for something that may be host and maybe similar to the virus, but it's a host gene, because then we would have nothing but a lot of false positives. So in combination, in not just in the biotechnology or the testing of it, but there are three concepts that have to be brought together in Koch's postulates, modified or not, that need to be looked at for a disease. We want to look at the epidemiology of the disease. Where is it occurring? What group of people is it occurring in? What part of the world is it occurring in? Are there animals present? What environmental factors are present? Did all of these people eat the same thing? Were all of these people in the same place? Those sorts of things. We also look at clinical observations. Do they have a fever? Do they all have a similar fever? We look at signs and symptoms. So we can get a combination, get an idea of signs and symptoms of a disease, and then you're like, well, you have the flu or you have COVID-19. Like, one of the issues with COVID-19 now is that there are so many early signs and symptoms that mimic other conditions like the common cold or the flu, that it's really difficult. I mean, you could just have hay fever and a cough, and now all of a sudden you think you have COVID-19. So there is that concern there with this particular virus that we're all dealing with, that in clinical observations, it can sometimes be mistaken for something else. So you might think you only have a cold when you actually could have COVID. And you go to the grocery store, you go out, and you end up exposing other people, even though that's not what you think you have. And that is the biggest concern, and why epidemiologists and public health officials want this quarantine. This last is your pathology and microscopy. This is the actual testing. So they would There's lots of testing where they're finally introducing those lateral flow devices we talked about in class a few weeks ago. They're finally starting to come out with that. I saw it in the news last week. So see, we were all like, we were months ahead of them. All right, so we have disease. So all three of these factors here, all of these factors are brought together to determine a specific disease. When we determine that disease, we look for something genetically, some kind of molecular signature. If we can find that molecular signature, this is that PCR test that we're looking for, and this process over here is how they find the molecular signature. Once we know this, we can now identify the agent. We can also look and see if it's related to other known agents, which is what they did. Because it's a coronavirus, it is related to MERS and SARS. These are both coronaviruses. And so COVID is related to these two. So not only were we able to identify it as its own, its putative pathogenic agent is what it means as the current. Not only do we identify it as pathogenic agent, but we also know that it's very closely related to SARS and MERS. And so we can definitively say that it's a coronavirus causing COVID-19, and that COVID-19 is a coronavirus, and we know that it's its own, so it's coronavirus D 2019. So that's where COVID-19 came from. So mechanisms of disease. How do these diseases make us sick? What do they do? Most of this is in relation to bacterial diseases, mainly because they are the most common and they are the ones we know the most about. So first, we could ingest a toxin, and through ingestion, we can either take in the toxin, the bacteria that's causing the toxin, but more often than not, we don't have the bacteria growing in our system. We have just ingested toxin that the organism has produced. And the most common example of this is staphylococcal food poisoning. And it's caused by Staph aureus, and it's why you have to wash your hands every time you work with food, as well as if you work in a restaurant or like a commercial kitchen type of thing. You have to wear gloves all of the time and hairnets and that kind of stuff. And that's all because we want to avoid, for the most part, staphylococcal food poisoning. Staph produces a really nasty toxin when it grows in, it'll grow in greasy or fat-like foods. It grows in foods that are very salty because think about where staph would normally grow on your skin. It can withstand a high salt environment. It also grows in foods that contain a lot of dairy. So cheeses, cream pies, those sorts of things. And this toxin that Staph aureus produces is also what we call temperature labile. So it can actually withstand low temperatures, like you would if you make a custard or something, some kind of creamy dessert where you have to warm the food, but you don't heat it up all the way. Like boiling, the toxins produced by Staph aureus can actually survive within that warmer than normal environment. So those foods can actually, and staph, by the way, can survive in those warmer environments too. So as long as the food is sitting out at a room temperature, then Staphylococcus will grow in the food. And during that growth, it will produce metabolic waste toxins. When you consume the food, you immediately are consuming high concentrations of the toxin. The bacteria is not what's going to give you the symptoms of food poisoning, it's the toxin. And this is why staphylococcal food poisoning occurs within hours after ingestion, usually 1 to 6 hours. So it happens very, very quickly. Another form of ingested disease or organisms to cause disease is colonization in mucous membranes. Now this occurs when, let's take salmonella for example. If you ingest a food that contains salmonella in it, salmonella will begin growing in your intestinal tract. It produces toxins that break down tissue, and those toxins and the salmonella itself are going to cause things like a fever, nausea, diarrhea, those sorts of things. So the bacteria begin actually growing within the system. So we have staphylococcal food poisoning because that's a toxin that's poisoning you, but salmonellosis or salmonella infection is actually considered a food infection, not food poisoning. So in that colonization of mucous membranes, of course, you have salmonella I talked about, but probably more virulent would be Vibrio and the E. coli, what we call this is from a group of E. coli's called EHECs. This stands for enterohemorrhagic E. coli, Escherichia coli. And 0157H7 is the most famous one. This is the one that we oftentimes have food recalls. Spinach, bean sprouts, alfalfa sprouts, a bunch of other foods are oftentimes recalled because of this E. coli 0157H7. It's very difficult to get rid of, and it's usually spread by wildlife. It grows in most warm-blooded mammals, and so this wildlife will get into fields where all of this produce is being grown, and then they'll defecate in those fields. And when they do, these vegetables here will get covered in E. coli, so it can oftentimes spread. Then we have another mechanism is known as invasion of tissue, where once the organism, the pathogen, gains entry into host cells, it will actually begin replicating. So Salmonella does this, TB does this, and both of these, as well as several others, have the ability to evade host defenses. As long as they're inside of the cell and they're replicating inside of our tissue, then our immune system is not necessarily always going to catch them early enough to get rid of them. So they may actually be able to replicate more rapidly than the immune system can get them under control. The last is invasion of tissue, and instead of just tissue toxin, instead of just tissue damage, we also have, in addition to tissue damage, the production of a toxin. And this is something Shigella and tetanus do. So like with a tetanus infection, we don't give, when somebody gets a tetanus shot, it's actually the toxin that we're giving the tetanus shot for to reduce toxin, not reduce toxin production, but to neutralize any toxin that may be produced by tetanus should we be introduced to it. Tetanus is super common. It's out in the soil. That's why everybody gets a tetanus shot. It's part of the vaccine schedule. And tetanus, when it grows inside of the body, begins producing the tetanus toxin, which is causes rigid paralysis and can be quite deadly. Now, adherence, the ability of a pathogen to be able to stick to and adhere to cells or host tissue is a primary factor in its ability to be pathogenic. These organisms, if they're just floating around and they're producing toxins, the toxins themselves will also have to adhere and gain entry into cells. So this whole adhesion ability to host tissue is really important. So some bacteria will produce adhesions, and these adhesions that are found are fimbriae. We've talked about those. Fimbriae are those long structures found on the outside. They're not cilia. They're just fimbriae, long protein-like fibers or hair-like structures. They're usually pili that have an adhesive property to them. They cause the organisms to bind to sugars on the cell membrane. So we have the cell like this, and our cells, all cells have, receptors on them, and they have these polysaccharides. They're kind of like tags so that these cells know. And what the bacteria have is they have fimbriae, and the fimbriae attach to the sugar molecules. And this allows the bacteria to then adhere to our cells. Here, I'll try and mute. Is that better? I muted everyone. Is that a little better? Okay, great. I'm just going to mute everybody. Okay. I couldn't hear it, so I apologize for that. All right. Then in addition to, that's okay, Rolando, you didn't know. All right, so in addition to adhesions, we also have colonization. Remember, bacteria can produce those siderophores. So here I have a bacteria cell, and siderophores are these little iron-binding molecules. And what bacteria do is they produce them, but they kind of have an affinity for the bacterial cell. So they're out here and they surround the bacterial cell. And then the iron, I'll just put iron in yellow, attaches to the siderophores. And then these siderophores will then get reabsorbed by the bacteria to bring the iron inside the cell. Also, some bacteria will produce IgA proteases, and these are enzymes that will break down IgA antibodies. If you remember, IgA antibodies are found in mucous membranes. And mucous membranes are the #1 portal of entry. That's how most organisms get into our system. So if you are a bacteria, or if you're a bacteria and you want to gain entry through a mucous membrane, it would be beneficial for you to produce an enzyme that would break down IgA antibodies, because then they cannot attach to you. If IgA antibodies attach to the microorganism, they will neutralize it. The organism will not be able to adhere to the cell and thereby won't be able to gain entry or cause disease. Then we have what are called effector proteins. Mostly gram negatives produce these, and these are called secretion proteins. And there are, I think, 4 different types of them. The one we're going to look at is a type 3. And a type 3 secretion protein will cause cytoskeletal elements to kind of to rearrange. Now, bacteria are kind of big. Think about E. coli. E. coli is a pretty decent sized cell. And it's still smaller than our own cells, so it can gain entry into cells fairly easily, but it has to get past that cytoskeleton. And our cytoskeleton is really thick and very intense. It's, you know, holding the shape of the entire cell. So in order to get past it, it's kind of like a chain link fence underneath the cell membrane. So some bacteria may be too large to get through that chain link fence. So they will produce these type 3 secretion proteins to open the cytoskeleton. I use the analogy, I always envision it as kind of a pair of bolt cutters, and you're trying to get into a park, or you're trying to get into a property and you can't get past that chain link fence, so you break out a pair of bolt cutters and you cut a hole in the chain link fence. And so those bolt cutters would be an injectosome. Once the pathogen gets into the cell, oftentimes that entry will cause some kind of damage. So if there's damage, then other organisms can gain entry. Also, if you damage your own cells, that also allows for entry, such as you cut yourself. And when you cut yourself, that now exposes your skin flora to blood and serum in a new environment that they aren't normally in. So they will then start taking advantage and become opportunistic pathogens. Mucous membranes are the most susceptible part of the body. That's one of the reasons our immune system puts so many IgA antibodies in the mucus membranes. One of the issues is here we're talking this example over here. This is in the lumen tissue in the endomembrane system of the gastrointestinal tract. And what we're looking at here are Peyer's patches, which we talked about in immunology, and you guys probably learned about in anatomy too. Does anybody remember Peyer's patches? You can just give me a little agree. Stephanie says no, Jalen. Stephanie, you probably had to identify it on a slide. Usually that's one of the structures in the intestinal lab they make you recognize on a slide. So in that slide, when you guys were looking at Peyer's patches, what we have here is kind of the idea of a Peyer's patch. So these are right here. Let me delete what's going on here. So these cells here are your endothelial cells. Where's my pen? These are your endothelial cells that are lining, right, the intestine, the inside of the intestines. Underneath them down here is your Peyer's patch. So this whole yellow area here is the Peyer's patch. Now, our Peyer's patches are constantly, they're part of our immune system, right? Remember, they're part of that MALT tissue, mucosal associated lymphatic tissue. They're part of MALT. And when these Peyer's patches, what they're doing is they're kind of sampling what's going on in the intestines. So they're forever sampling and presenting and creating. This is where IgA antibodies are primarily made. Now, what happens with some of these organisms is in order for the Peyer's patch to be able to sample what's going on, every couple of cells in the lumen, there's a specialized cell called an M cell. The M stands for microfold. This will be a cell that has no cilia. It's non-ciliated like the other cells. It's kind of flat. And bacteria and other pathogens can enter across the lumen through these M cells. Now, the Peyer's patches do this on purpose because they want to sample what's going on inside of the gastrointestinal tract. So that part of the immune system is like, hey, what's going on in there? What's coming through? It's kind of like a checkpoint, like checking all of the traffic. Now, some organisms have evolved a way to avoid this process, and one of them is Shigella. So Shigella will enter through an M cell with the function or the purpose of getting phagocytized, which is what happens in a Peyer's patch. But instead of being phagocytized and destroyed, the Shigella bacteria produces a toxin that allows it to break out of the phagolysosome and out of the macrophage and enter through this kind of back channel into the endothelial cells in the lumen of the GI tract. It then moves from one cell to the next, and in doing so can transfer itself all the way through millions of cells in the GI tract. As it does this, it starts to destroy these cells. So the ciliated lining of the gastrointestinal tract will start to shed. Those cells all start to die. And when they shed, and we have a lot of cell damage, first it will be severe diarrhea, followed by diarrhea that's strictly just water because there's no solids left in the GI tract to pass. So there's a ton of water being wasted out. Shigellosis is a pretty yucky disease. And then eventually it'll turn into sometimes referred to as rice water stool. And this is where the cell debris is starting to be released with the fluid in the diarrhea, and it looks like grains of rice, small grains of rice. And that is cell damage. Those are dead cells that are being shed from the internal lining of the GI tract. So this is how just through entry, there can be damage to the cells. Again, mucous membrane susceptible. Some bacteria will specifically direct endocytosis. They want endocytosis to occur either in antigen presenting or phagocytic cells. Sometimes they'll do this in non-phagocytic cells, and this causes a condition known as membrane ruffling. Another is that antigen sampling. That's what I was telling you about here with Shigella, and that's using M cells to enter into Peyer's patches is just one example. But many organisms will purposely be phagocytized so they can then move out of that phagocytic cell to get somewhere else. And they'll move from one host cell to the next host cell directly. Listeria does this as well. And they'll move from one host cell to the next so they won't be detected by the immune system. Is everybody with me so far? Do I have anybody who has any questions? Feel free, just raise your hand if you have a question. Okay. If you have a question or you don't understand something, raise your hand and let me know that so I know to stop. Okay. So we're going to now look at avoiding the host defenses. What do these organisms do to avoid host defenses? Many of them have evolved all kinds of different mechanisms, and it's really kind of fascinating. We have hiding within the host cell, which we just looked at. Listeria, Shigella. Listeria will rearrange the actin filaments inside of a cell and use those actin filaments to propel itself into a neighboring cell, which is really just kind of blows my mind. Listeria takes a very long time to show up. It has an incubation period anywhere from 3 to 8 weeks. It is a gastrointestinal disease. It's usually it's a food infection, but it takes a really long time to show up. It has a fairly decent mortality rate considering other food infections like Salmonella or even Shigella. Listeria is very complex disease. And it's very difficult to diagnose because it's transmitted through ingestion. So figuring out what somebody ate 3 to 8 weeks ago can be very difficult unless it's someone that keeps track of their food. Or most often we see outbreaks of it in places like a nursing home or a assisted care facility, and those are determined usually because those facilities keep track of what all of their patients eat over time. Then we have something called serum resistance. This is where an organism is able to avoid the complement system, and they usually do this by producing some kind of toxin that breaks down complement proteins. So they produce, it's a class of proteins called proteases. And then they can either prevent, survive, or avoid phagocytosis. And that's a big one because phagocytosis is really important for initiating the immune system. So if these organisms can avoid it or prevent it at all, then the organism can kind of grow unchecked. If we don't activate that adaptive immune system, then we run into a loss of checks and production of antibodies and other factors that work best to get rid of organisms. So we have what are called C5a peptidases, so avoiding complement again, right? Breaking down C5. If we break down C5, this will prevent receptor-mediated endocytosis. So it prevents, anybody remember that term? Receptor-mediated endocytosis. What do we call it when we enhance phagocytosis? Anybody remember that term from last chapter, last unit? Yes, opsonization, exactly. So if we can prevent opsonization, then, because C5b, remember C5b was what attached. So C5a, this will prevent the inflammatory. So this prevents inflammation, which recruits all of those phagocytic cells. And when it destroys C5, it prevents C5b from binding. And if C5b can't bind, then this prevents opsonization. My handwriting's terrible on here. Sorry, guys. So and in this case, with the C5a peptidase, I can prevent both a very strong, remember, C5a is a really strong anaphylatoxin, so I can prevent one of my primary inflammatory signals as well as prevent opsonization. So I'm preventing inflammation, I'm preventing phagocytic cells from being recruited heavily. In addition to those that do finally get there, I can prevent them from having receptor-mediated endocytosis. So I can reduce the phagocytic activity. So if I greatly reduce that phagocytic activity, in turn, I'm reducing antigen presentation, which in turn reduces antibody production and activation of my adaptive immune system. The last one is membrane damaging toxins. These guys will be phagocytized and then actually destroy the phagocytic cell in the process. Other ways that host defenses are avoided is through capsule formation, binding complementary regulatory proteins. This causes a shutdown of the complement system so that it just doesn't respond. This is done by what's in Streptococcus. They produce an enzyme called an M protein, and the M protein reduces or turns off complement. Then we have what are called FC receptors. FC receptors are little peptides that bind the constant region of antibodies. So let's see what that looks like. Here I have my bacteria, right? And my bacteria have little protein-like structures on them that they use for cell signaling or attachment. Maybe these are fimbriae or pili or whatever the case may be. But these are antigenic determinants that are found on the surface of these bacteria. So when we produce antigenic determinants, when bacteria produce these antigenic determinants, we produce antibodies against them. So antibodies start attaching. And we've got antibodies that attach to these. And can anybody tell me, if I have antibodies attached to something, there are 6 effector functions of antibodies. There are 2 of them that involve other cellular responses. And aside from complement activation through classical activation, I can also have antibodies recruit phagocytic cells. So antibodies neutralize, they cover up this bacteria, the bacteria gets covered, but this bacteria may be producing toxins that are making it easier for other bacteria to gain entry, say, into a cell or something like that. Now, this bacteria doesn't want to die. It wants to reproduce. It wants to divide. So it's going to produce its own set of little proteins that will attach to the constant region of all of these antibodies that are attached to it. And in doing this, the phagocytic cell can't use RME, can't use receptor-mediated endocytosis, because the receptor that this would bind to won't accept this little cap on the end of it. My natural killer cells are not going to recognize it or be able to attach to it. My cytotoxic T cells, or my CD8 T cells, are not going to be able to bind to it because the receptor for that is now hidden as well. So by covering up the section of the antibody that the immune system would recognize, this bacteria can now survive. Everybody with me still? Okay, great. How do these organisms survive phagocytosis? To survive phagocytosis, one thing they do is they escape the phagosome. So remember, you have the 5 or 6 stages of phagocytosis, and one of them was the phagosome, right? So here we have our big giant phagocytic cell. So here's the phagosome, and then we had the lysosome, and these two would merge together to become the phagolysosome, right? That's what's going on here. So if these guys get ingested, they'll purposely get ingested. They'll be inside of the phagosome, and before this merge can occur, they will escape. And then they are now an intracellular inside of the phagocytic cell, but they are not going to be merged with the lysosome or destroyed. They can simply just prevent this fusion reaction from occurring so they don't get destroyed. Usually they'll do that and then escape. Or they can survive in the phagosome. Now this one's really crazy, but here is inside of a phagosome. And when a phagosome merges with the lysosome, I'm not sure if we covered this, but did we cover how a lysosome works? Didn't we talk about that this term? Anybody remember? Briefly, okay. So I'm going to go and share the whiteboard. I want to show you how a lysosome functions real quick, and I'd like to use the whiteboard to do that. So I'll go to the whiteboard. All right, so I'm going to draw my, here's our phagocytic cell, right? So here's the phagocyte. Doesn't matter what kind it is. We'll just put it as a phagocyte. And inside of the phagocyte are these lysosomes, right? I'll just give them the letter L. And we have lots of lysosomes. And when a phagocyte merges, when it takes in, right, a bacteria like this, that bacteria attaches to it, and then this guy will create kind of an indent like this, and that bacteria will be taken in here. And so this is our phagosome. And what's going to happen is that lysosome then fuses with the phagosome, and it all becomes that phagolysosome. Now, a lysosome works like this. Our bacteria gets taken in, and we're inside actually a phagolysosome here. And when a lysosome membrane fuses with a phagosome, it actually turns on proton pumps. So I'm going to put them right here. So these are, I'll make them circles just to make them easier to see. So these are proton pumps. And what's going to happen is this lysosome begins pumping protons into this phagolysosome. So all these protons start building up inside of the phagolysosome. And when we increase the number of protons in an enclosed environment, what happens to the pH of that environment? What happens to the pH of an environment? Does it go up or down? Does the pH increase or decrease if I add protons to it? Correct, it does. It goes down. And if the pH is low, that means that my environment is acidic or alkaline. Correct, acidic, right? So it lowers the pH, making it acidic. Now, inside of that lysosome, and what makes these lysosomes function is a set of proteins, right? So I'm going to make these little proteins like this. So I've got these little proteins here, and those proteins are called acid hydrolases. And what these enzymes do is when an environment becomes acidic, they will hydrolyze whatever is in there. But they will only do it if the environment is acidic. So when this bacteria here gets brought in and this phagosome fuses with the lysosome, this turns on the lysosome proton pumps. This fills up with protons, thereby lowering the pH. The pH becomes very acidic. In an acidic pH, my hydrolases get activated and they start destroying the bacteria or anything. Whatever is in there, they're going to break it down. They hydrolyze it, right? Remember hydrolysis? So any organic molecule that's taken in here, they're going to be able to carry out hydrolysis and break it down. Now, the reason for this complicated activity is because lysosomes are in all of our cells. And if lysosomes were to be damaged or break open in the process, we want to make sure that those hydrolase enzymes are inactivated in a neutral pH. They won't function when the pH is neutral, which is what we have in our cytoplasm. So if this were to break open by accident and release these enzymes into the cell cytoplasm, those enzymes would be inactivated. They'd be denatured because they can't function at a neutral pH. They can only function at an acidic pH of around, I think it's around 3 or 4. So it has to be really acidic inside of that lysosome. You could say that, Brittany, yes, but they don't explode like proton grenades. There you go. Yeah. And what's really kind of interesting about them is bacteria, that what happens with the bacteria is now my bacteria that are in there, my bacteria start releasing OHs. And what happens to the pH when we start releasing OHs? Hydroxyls. What happens to the pH? It goes up. So I have my lysosome pumping protons in to try and lower the pH, but I've got my bacteria releasing OHs. So they release compounds or buffers is essentially what they're doing. So they'll release some type of phosphate buffer or hydroxyl groups alone. This in turn will neutralize the pH inside of the lysosome. So what happens to my acid hydrolases? Are they neutralized or are they still functional? Correct, Victoria, they get neutralized. And if they're neutralized, then my bacteria is now free to survive or even break out of the lysosome. So in this case, the lysosome will have no effect. It won't be able to break down the bacteria. And so all the organism has to do is release some kind of alkaline product to neutralize the pH inside of the lysosome so that those acid hydrolases will not break it down. When we studied the viral chapter, we saw that activity, that pH activity, when we looked at the dengue virus infection video, and they showed how the acids inside of the lysosome, once the dengue virus was entering into the lysosome, the dengue virus actually used that acidic environment to cause denaturation of capsid proteins. And that denaturation resulted in a structure that allowed the dengue virus to exit into the cytoplasm. So in that case, the virus was purposely being phagocytized so it could use the phagolysosome of cells, of just regular cells. It wasn't even phagocytic cells. So that it could use that lysosome to break out into, to get rid of its, that outer protein coating, so gain entry into the cell to take over. Everybody good? Great. So I'm going to go back to our PowerPoint. I just gotta find where they are because it takes me back to the beginning. There we go. So that's that whole escaping from the phagosome after ingestion. You can see by preventing fusion, then it would prevent that lysosome, those enzymes from being able to destroy anything. And of course, surviving in the phagosome, that would mostly be through neutralizing compounds against those digestive enzymes. So here's an example of Listeria. I told you guys about Listeria gaining entry. So here Listeria has gained entry through phagocytosis into a host cell. And here it is inside that phagosome, and it escapes out of the phagosome. Now once inside of the cell, what this bacteria does is it starts to grab actin microfilaments inside of the cell, and it starts attaching them to what's known as a tail assembly. And it takes our own actin filaments inside of our cell and assembles them, attaches them to its surface, so it can then use them to push themselves. Now look, it's using our membrane. And so it fuses the membrane of this cell to the membrane of this cell, bringing it inside through engulfment, right? Because our neighboring cell says, oh, look, my neighbor's giving me something. And so it accepts what the neighbor cell is giving it, and it's not going to phagocytize it. And this is how Listeria can move from one cell to the other and completely avoid the immune system. This whole actin tail assembly and the life cycle of Listeria is very, very slow. So this takes a long time to occur, and this is why the incubation period, time of exposure to time of signs and symptoms, that incubation period is so long. This electron micrograph over here, this fluorescent microscopy actually, the actin filaments are green, right? So we have the bacteria are red. You can see there's some of these actin filaments here. I'll erase that. And the bacteria here are red. And then these actin fibers, you can see these are all kind of green. So they're creating these filament tails, and they're going to start moving around. Look, he's moving from one cell to the next. So now the tail's kind of broken down as he moves from this cell into that cell. We've got a couple of them showing up here too. So as they start moving and creating these actin filament tails, we can start to see them. Antibody evasion. How do these guys stay away from antibodies? This is what we call antigenic variation and shift, and this is what's going on right now with COVID. So we're going to talk about this, and then we'll be done for today. But I want to talk about a few other things on this slide. First off, using proteins to break down antibodies. We talked about the IgA proteases in mucous membranes. Luckily, there's not too many IgG or IgM. We mostly see this with just simply against IgA antibodies. The other is the cloning of host molecules. Some bacteria will either coat themselves in host molecules or produce their own molecules that mimic our own. Some bacteria, like Streptococcus, their capsule, remember these are capsule-forming bacteria, they'll add hyaluronic acid to their capsules, and this is part of our connective tissue. So when our immune system sees hyaluronic acid, it thinks that it's our own cells and not so much a capsule in strep, and this is one of the reasons that strep can avoid the immune system so well. But what I want to spend some time on here is antigenic variation. Antigenic variation is kind of what we're going through right now with this COVID virus. It's really kind of an interesting concept, and we're seeing it more and more, especially in viruses, but it can also occur in bacteria and some parasites. It was actually first discovered in trypanosomes. So what happens in antigenic variation? I'm going to start with a drawing on with just antibodies, and then we'll look at this diagram on the left. So let's say that you are infected by a pathogen, and your body produced antibodies against it, which is our kind of normal reaction. So here we have all these antibodies that we've produced against that pathogen. And so I have all these pathogens as it replicates. These pathogens all have the same antigenic determinants. But somewhere along the way, one of the pathogens produced was a little bit different. So when my antibodies get rid of all of those antigenic determinants, the only thing that's left are now my new bacteria. And these new bacteria are different. Now, eventually, my body's going to produce antibodies against it, and they're going to stick to it. So I start producing antibodies, and I start getting rid of all of these bacteria. And my bacteria, all of a sudden, I get a little bit of a change. As all of those bacteria get destroyed, the only thing that's going to be left are the ones that I didn't produce antibodies against. And so this just keeps going on and on and on and on. Now, in viruses, we see this a lot. And in viruses, this is what's known as antigenic shift and antigenic drift. So antigenic drift is our annual flu virus. This is mostly seen in flu viruses. Okay? So we have our annual flu, and this is why we get a flu shot every year. So let's say I got my flu shot this year. My flu shot this year is specific against whatever version of flu is out there. And so this evoked my body to produce black antibodies and pink antibodies against the black and pink antigenic determinants of this year's flu virus. Now, flu viruses, because there are so many of them and they are so interspecies, they kind of move around between species often, and you can be infected sometimes even by more than one flu virus or be exposed to more than one type of flu virus at the same time. And when this happens, we get what's called antigenic drift. And every year we come out with a new flu vaccine because this next year, my black antibodies here might still function against the black antigen that's present on this new version of the flu. But over a year's time, my flu virus switched from a pink antigen to a blue antigen. And this blue antigen, I don't have antibodies against. So although my flu vaccine from this year may be effective against flu next year, it's not as effective because now my flu virus has a new antigen that my body didn't see before. And so I still have some immunity, right? I still have these black antibodies against it, but those black antibodies are not going to protect me against the blue antigen. And so now that I have this blue antigen, I'm going to have to create antibodies against that blue antigen, and I would rather do that with a vaccine than actually get the flu. So the flu vaccine from last year will give you some immunity. It's not totally ineffective, but it's not as efficient or effective. It has what we call low efficacy against the current flu virus. And this is because flu viruses go through this antigenic drift. Okay, so an analogy for this would be imagine every day to class, I came into class and I was wearing a purple cowboy hat. Every single day. Every time I came to class, I had on a purple cowboy hat. And you got so used to that purple cowboy hat, you so recognized it that if you were walking into the library on a Friday afternoon and you saw a purple cowboy hat on the other side of campus, you would be like, oh, look, there's Professor B, because you recognized that purple cowboy hat. So you would always see that. Now, one day I surprise everyone and I'm on campus and I have a yellow cowboy hat. Now, you might recognize the cowboy hat and say, wow, look at that crazy colored cowboy hat. Professor B always wears a purple one. I wonder if she switched, and that's her. She's wearing a yellow one. So you would recognize me, but you're not so sure that it's me because you're used to seeing purple, and this person's wearing a yellow one. It could be me. It could not be me. You're not sure. If it were purple, you would be for sure that that it was me. I'll answer your question in a second. Now, and that's antigenic drift. So you see it and you recognize it, but it's not entirely, you're not entirely sure because of that shift. Now in because of that drift, excuse me. Now in antigenic shift, what happens is we have flu, usually from multiple species. So we have a pig flu and a human flu, and pigs are this mixing vessel because for some reason, pigs can accept bird flus, they can accept human flus, they can be infected by their own flus, and some other warm-blooded mammals like bats and those sorts of things. So pigs have receptors and have the ability to be infected by a multitude of different species of flu viruses. When that happens, these flu viruses mix If we get a single pig cell infected by a human flu, a pig flu, and a bird flu, the genetics of those three different viruses, in this case of these two different viruses mixed together, what happens is we come up with a completely new virus that none of my antibodies are going to be effective against. So this is referred to as a novel virus. And a novel virus is one that we have no protection against. This is what happened in the H1N1 outbreak, which was a pandemic years ago. Now, the reason that H1N1 was not as severe was because we already had flu vaccines. So we were able to produce a vaccine against the flu, against the H1N1 flu in a matter of about 2 months. They just sped up the current flu, and now every flu vaccine you get includes the H1N1 antigens. So the flu is a pandemic every year, but it's not one that we're constantly battling or quarantined against, and that is because we have some herd immunity and we have vaccines against it. With COVID-19, we don't have any of that. There's no antigenic drift. It's a complete antigenic shift of a completely different class of viruses, which is the coronaviruses. And we don't have vaccines against coronaviruses. The first coronavirus to really cause any major concern, coronaviruses are usually just common colds. But through mutation, the first coronavirus that caused a really severe, deadly respiratory infection was SARS. And SARS was really not, 2010? So that really was not very long ago. After SARS came the Middle Eastern respiratory syndrome, so that's MERS. Both of these are coronaviruses. MERS was an antigenic shift. Now, antigenic shift in the flu takes about every 10 to 15 years. We see some kind of antigenic shift in a flu virus. Antigenic drift can happen every 1 to 2 years, right? Because it's just a mild change. This is an entirely new virus, and it normally takes a long time for that to happen. SARS in 2010, MERS in I think around 2014, 2015, that's half the time. And now in 2020, we have COVID, and all of these are coronaviruses. So the antigenic shift that's occurring in the coronavirus is occurring very rapidly. There's a lot of mutations occurring in that coronavirus. On top of that, the transition, what we call the jump the species, so the jumping from one species to another, that speciation, there was no pig. That jumped from one warm-blooded mammal to another. Now, usually there's some kind of intermediary. There's a few species jumps that have to occur first, like bird to pig to human. But there was no pig involved in the coronavirus shift. So this shift is occurring every 5 years, in half the amount of time that we would normally see in a flu virus. And that is of another concern that really no one has addressed much yet, at least not publicly. It's in some of the papers I've been reading, some of the updates I've been reading. There's wow, this shift is really occurring very quickly. But part of that is it's believed to be more of a drift, an antigenic drift, but the problem is we don't have vaccines against any of these. There are no vaccines against SARS, none against MERS, and none against COVID. All right, so I want to take questions. We're going to stop here lecture-wise for today. I want to answer a few questions. Jalen, does that mean that someone can get COVID twice if the virus is mutated? Well, I don't think they have determined that yet. I'm just going to be honest with you. There's so little known about this virus so far. They do know that it's difficult to come up with a vaccine against. SARS there was a big push to create a vaccine against SARS, and then that was kind of put on the back burner because the SARS epidemic went away. And so there wasn't as much, when there's not a need for it, companies that develop those things don't really push to continue developing it, so it was kind of shelved. It, I think, resurfaced again, the idea of a vaccine resurfaced again when MERS came out, when they were talking about MERS, but then it never occurred. I'm going to send you guys a link. Last Friday, the college had a town hall with a public health and epidemiologist from FIU, and she gave an incredible, it was really an absolutely incredible lecture on the virus. I'm going to send you guys a link to it. I really, really urge you to watch it. because it was all about COVID and all about like the vaccine development, the mechanism of the disease, the epidemiology of it, and it also has a whole lot of you're going to learn how to do an R factor, which we're going to talk about in our chapter, and the epidemiological curve, also about the serostatus and transition status of viruses and those sorts of things. So a lot of things that we're going to learn about in our epidemiology lecture, she actually talks about with COVID. So I think it's a really relevant lecture for you guys to watch when you get a chance. I'll put a link to it in D2L for you. Deja, I wouldn't say they sweep it under the rug. They just change priorities, right? So think about it. We have a limited number of resources. So the NIH would be ones that are working on this, but SARS kind of it was brought under control. There's not a lot of cases of it anymore. It's not something that's a worldwide concern. So the priority to create that vaccine was shifted towards something else. There were other diseases that were of greater concern at the time, right? We still have malaria. We still have flu killing thousands of people, right? So we still have other diseases that are much more prevalent. So I wouldn't say it was swept under the rug. I don't want to make it sound like they just shelved it because it wasn't worth it. But there were just other priorities that resources have to get shifted to. We can't do it all at once. Does anybody have any other questions? I gotta say, I know this sounds really, really weird, but I think that it's going to be interesting that we're going to be studying epidemiology and pathogenicity and all of this particular unit during this time. I think it's just really relevant, and we'll be able to determine a lot and learn a lot in this unit, in the situation that we're in. I know that sounds terrible. I don't want to be in this situation either, but we're in it, so maybe we can use it to our advantage in that sense. Does anybody have any other questions on pathogenicity? Yes, Jada, it's definitely a real world example. Exactly. Yes, very much so. All right, so I'm going to end the recording here. We are done for today. I will put this recording up as soon as Blackboard renders it. Don't forget to look in D2L. I put up a, you know what, Jalen, I'm going to go over that in the next lecture because we'll finish this unit up real quick and then we'll go through the case study. Yeah, we'll do that Wednesday. So I'm going to go ahead and finish this recording. Please make